Let's start off with part one, AMR, a perfectly natural phenomenon. And as most of you will know, antimicrobial resistance is pretty much just the description of the ability of a microbe to resist an antimicrobial substance. Those antimicrobial substances can just be in nature around them, or they might actually be secreted by other microbes. And it's now thought that antimicrobials and antimicrobial resistance very much form part of a microbial competition, communication, evolution. So AMR is just a normal part of antimicrobial of microbial life on a planet where different selective pressures abound. And it's not just something that happens slowly over the course of millennia, as we might sometimes think of evolution taking place, but because microbial life is so fast, microbial resistance also takes place at awe-inspiring speeds. Some of you might have seen this experiment published by Harvard a couple of years ago, where they built a giant petri dish and they let colonies of E. coli on both sides of this petri dish overgrow it and then hit various boundaries of antimicrobial selective substances. So the first line here is then, I think, one concentration that kills them, and then by the 1000th line, the concentrations have increased. And what you can see here really in this 11-day time lapse that I'm showing you is just how incredibly fast evolution happens, how incredibly fast microbes adapt to antimicrobials, and also that this is not just something that happens very rarely, but as you can see that this is happening almost in parallel on both sides of the petri dish. This adaptation happens across, it's a normal fact of microbial adaptation to different selective pressures. And, to perhaps nobody's great surprise here, by the end of these 11 days, the E. coli on both sides of this plate have managed to completely overgrow the plate with different evolutionary pathways later identified via the genomic study of this plate. So AMR is normal, AMR is very fast. And what does this mean for antimicrobials, the antimicrobials we use in human medicine and food production? Well, the answer is resistance is pretty much inevitable. Indeed, some studies think that any antimicrobial that will ever be discovered already has AMR factors circulating for it already at this point around the globe. So any antimicrobial we use will inevitably lead to, a gradual selection for microbes that are resistant to its effects. And this problem, this ability of microbes to resist the antimicrobials we use to control them, has over the last decade really led to increasing warnings at the global level that it might actually constitute something of an apocalypse. And one person who's been particularly vocal in using this vocabulary has been the UK's former chief medical officer, Dame Sally Davis, now the UK's AMR envoy, who has consistently been warning that we might be facing a time when the drugs, which with this she means the antibiotics, don't work, and the rise of antimicrobial resistance might lead us back to a pre-antibiotic era when human health systems fail and agricultural food systems no longer work. Now, Davies, when you interview her, is actually a lot more nuanced than these titles might imply. So she also doesn't say that the apocalypse will lead to complete systems collapse all at once, because other forms of infection control exist, and some drugs will continue to work. It's very rare for microbes to develop pan-resistance against antimicrobials, but the scenario is nonetheless fairly dire, with in 2019 already about 5 million deaths associated with AMR being estimated by the Lancet, and anthropologists and social scientists increasingly comparing AMR to a major infrastructural challenge for health and food systems that have become dependent on easy access to affordable antimicrobials. And when these antimicrobials no longer work, it's almost akin to the collapse of other essential infrastructures we depend on. We might think of water, gas, electricity. If these don't work, the health systems and food production systems also don't function. So it is a major systems challenge, and the systems challenge is primarily being brought about by our large-scale dependence on antimicrobials, which has emerged over the last 100 years. So dealing with AMR, the biological phenomenon, from a health systems perspective, actually automatically entails dealing with the historical and social sciences, or the historical and political economic dimensions of the antimicrobial infrastructures we've created. We can only explain our vulnerabilities if we look back at the social political history of the health infrastructures and food production systems that we've created around the globe since the 1950s. So, how old are these infrastructures? Now, I'm giving you a visual clue here behind me here, which is usually the clue that is most strongly associated with the founding legend of the antimicrobial era, right? I'm showing you here a moldy melon, and the reference here to mold usually leads directly to people answering, well, it's Alexander Fleming, right? So usually people associate the start of the antimicrobial era with Fleming and his rather unhygienic research practices in a laboratory in Queen Mary's Hospital in London, when a petri dish of his got overgrown by a mold culture, and that mold inhibited bacterial growth. And indeed, the Fleming myth has become very persistent around the world these days. Streets are named after Alexander Fleming. The UK's most prominent foreign aid program, the AMR, foreign aid program, is called the Fleming Fund, and you have plaques commemorating his actions all around the world. However, even though Fleming obviously did discover the effect of Penicillium notatum on bacteria, he was far from the only researcher during his time working on antimicrobials. Indeed, he was already conducting his research 28 years really into the rise of the antimicrobial era. Whereas Fleming now serves as a very nice figurehead for antibiotic engagement talks, the antimicrobial era itself had actually already started 20 to 28 years earlier in the laboratories of Central Europe, most prominently Germany during this time, when increasingly large teams of microbiologists and chemists started coming together to look whether they could find substances that specifically inhibited or killed bacterial cells while leaving human cells unharmed. And by the time penicillin came along, multiple compounds had already been discovered and brought to the market that had distinct antimicrobial effects and were being used across the world, ranging from compounds initially based on arsenic against sleeping sickness and against syphilis, to by the 1930s, the rise of the first kind of mass antimicrobials to reach the market, the sulfonamides, which come out of the discovery of Gerhard Domark in 1935. And then from the 1940s onwards, we've got a succession of new blockbuster antibiotics, so substances produced by microbes themselves, coming to the market and complementing this increasingly robust arsenal of chemical and biological drugs, really, that are being used to control microbes. And what's important here is not just that the antimicrobial era is older than the Fleming myth, but actually what the process of discovery tells us about how antimicrobials are already being perceived and conceived of by the scientific innovators. So rather than this Fleming story of a lone scientist, having a genius discovery, sitting alone by himself in a laboratory, what antimicrobials really result from is a large-scale effort, both by the part of industry and states during this time, to really get on top of microbial infections. So, be it Salvarsan or be it, the Sellman-Waxman's development of streptomycin, antimicrobials in the first half of the 20th century result from a systematized, large-scale research effort, both by the state and by private companies, to tackle microbial infections. And they're a product of big science, big industry, big states, and they result from actually quite diverse public and private R&D models. And the reason these models are so big and is being scaled up to such an extent is that states are desperately looking for ways of controlling microbial infections in increasingly well-subsidized welfare and medical systems. So antimicrobials here are already being developed with an eye to a potential market and application in increasingly sophisticated healthcare systems. So this isn't just, somebody having an invention by themselves sitting at a desk, but this is the result of actually quite a productive research system that is taking off and developing a whole range of new products by the end of the Second World War. And again, here, I'm just listing several of them. You can see the first arsenic-based products coming to the market from around 1905 onwards. By the 1930s, the sulfonamides come. The 40s is the era of the biological antibiotics. And already by the late 50s and early 60s, we start getting to an era of semi-synthetic antibiotics. And in many ways, this is the result of a much more systematized gaze on the microbial world on the part of science and industry. And you can see it here beautifully depicted in this picture by Robert Thorne, an American artist who was commissioned by Sharp and Dome to develop pictures that could be hung in physicians' offices all across the US in the 1940s and 1950s. And he shows here how this female microbiologist is screening for, antimicrobial compounds. She's overlooked here by an image of Alexander Fleming here. So, a bit of, the male patriarchal gaze here looking down on the female scientist. And then the product she finds will be upscaled in these deep fermentation vats. So this is very much is not a kind of chance product. It's part of a systematized, large-scale upscaling of a very systematized mode of looking through the natural world at this point. And the resulting panoply of new products that hit the market lead to dramatic changes in the way medicine is conducted. Often when we think about antibiotics, we think about the kind of heroic narratives of penicillin or another antibiotic or antimicrobial bringing people back from the dead or curing previously untreatable infections. But it's also worth considering all of the new forms of medical interventions that become possible on antimicrobials' backs. Think of open bowel surgery, for example. It wouldn't have been possible without modern antimicrobials coming along. Modern chemotherapy against cancer, keeping patients in immunocompromised states for so long, only really becomes possible because you have cheap, affordable antimicrobials that enable microbial infections to be controlled. In a large part, the healthcare systems that they developed in the 50s and 60s, think of these new big hospitals with wards that allow patients to circulate, very distinct from the previous isolation wards of previous eras. All of this is made possible on the back of antimicrobials, which not only enable new techniques, but also make the care and the control of microbial infections much more affordable. If you have antimicrobials, you don't need to invest so much in space to isolate individual patients, right? So you can develop much more affordable, much broader health and welfare systems. The antimicrobials here, they almost become infrastructural, right? These hospitals here, they wouldn't be able to exist as efficiently, as effectively as they do in the 50s, 60s, and 70s without all of these cheap new wonder substances coming along and enabling their smooth running. And, I would be remiss to say that this is only in human medicine. Indeed, animal medicine adopts antimicrobials almost as quickly as clinicians do in human medical settings. From the mid-1930s onwards, sulfonamides already get used to treat high-value individual animals, such as cows or horses. And by the 1940s, with specifically the Allies facing big constraints on their animal production during the Second World War, antimicrobials get used increasingly to medicate entire herds. Initially, this is done metaphylactically, so if one animal gets sick, you medicate all animals in a flock or in a herd, so as to meet your production goals. And then by the late 1940s, this is also done prophylactically because you can usually infections occur at specific times in an animal's life cycle. So for example, of a new flock being brought into a new building, at this point, it makes sense to medicate animals prophylactically to prevent infections from spreading in the first place. So antimicrobials here, they become also infrastructural in terms of facilitating ever more dense concentrations of animals living together during this period. And by the late 1940s, it's even surmised that antimicrobials might optimize animals' microflora and also treat subclinical infections so as to optimize their growth. And this is conveniently allows the antibiotic producers to offload their fermentation waste with very low residues of antibiotics into animal production. And you can see the result behind me here in this wonderful kind of collage of 1950s advertisement posters. The hope here is very much that antibiotics will do the same as they're doing for medicine, which is to make farming scientific. The farm is no longer a backward business, something where income is variable, but thanks to antibiotics, animals reproduce reliable profits. Thanks to antibiotics, this little pig here will no longer be a business liability, but will almost, as on a factory line, produce a profit thanks to modern science. And you can even see this here being heralded on this Pfizer poster where science is coming to the farm in a feedback. And this is so popular that by the mid-1960s, the majority of antimicrobials marketed in the US are no longer given to humans, but actually given to animals with animal feed additives, having a very large proportion of the overall antibiotic market in the US. And it's not just animals, right? Antibiotics also enter large areas of crop production during this period from the 1950s onwards. They're used in forestry. In the US context, they're also used in fruit trees, in sprays and injections to combat bacterial infection. So tens of thousands of kilos of substances such as streptomycin, etc., are used in these settings. And in the 1970s and 1960s, industrializing Asian countries such as Japan introduced a whole new genre of antimicrobials, such as kasugamycin, for example, to treat rice blast disease and enable intensification of rice production in these areas. So antimicrobials again assume an infrastructural dimension here. And perhaps finally, and most bizarrely, for a while, antimicrobials were also seen as the solution to global food shortages in the sense that they would enable a significant expansion of food preservation in areas where no refrigeration was available. So in the 1950s, oreomycin dips, tetracycline dips essentially, get licensed for poultry and for fish preservation across the Americas and in also parts of Europe. And the idea is very much you just kind of dip a raw poultry carcass or raw fish into these solutions. And because these antimicrobials will retard bacterial spoilage, your food becomes available for longer periods of time. You reduce waste and you overcome the global feared protein gap, which was being projected by demographers during this time. In the UK context, antimicrobials get included in the ice of trawlers that are being sent out into the North Atlantic to keep them on the water for longer, so the fish doesn't go off, the trawler can fish for longer, and its efficiency increases. And perhaps most bizarrely for a while, antimicrobials also seem to herald a new takeoff of the global whaling industry, with the Soviets, the Canadians, the Americans, and also Icelandic whalers incorporating antibiotics into harpoons, which you shoot at a whale, they'd explode on contact with the whale, release antibiotics into the dying animal's circulatory system, and thereby preserve its meat. It's notoriously difficult to preserve whale meat because whales are so big, they take such a long time to cool down that often the meat's gone off by the time you can actually start processing the animal. So there was a hope that thanks to antibiotics, whale meat would become a new abundant staple. Now, this is clearly the most bizarre example of antibiotics really kind of conquering all areas of food production, but also medical applications. But it just goes to show that what antimicrobials seemed to herald to people at this time was that thanks to this systematized big science on the part of states and on the part of industry, microbial control could kind of be expanded without limit. That there were no longer any limits by the microbe imposed on human industry and on human medicine, and that thanks to antimicrobials, all of these things would also become economically viable and more efficient because you could replace human labor that had previously been involved in controlling microbes and thereby achieve these highly efficient health and welfare systems, which became a hallmark of modernity. So one could almost say that antimicrobials, again, are the infrastructure underpinning large parts of the modernity's promise on both sides of the Iron Curtain. So no side can fulfill its promises of modernity to its populations without access to antimicrobials. But as I already mentioned, right, every use of an antimicrobial comes at the cost of inevitable resistance selection of microbes. So every infrastructure you build with the help of antimicrobials, sorry for this kind of rather crude metaphor, is built on crumbling foundations. Everything will inevitably lead to resistance selection. And it does beg the question whether people who were allowing these substances to proliferate in such an incredible way during the 50s and 60s did not know about AMR. And the answer, perhaps to our great surprise, is that they actually did know about AMR. Indeed, reports about AMR, perhaps also unsurprisingly, are almost as old as the use of antimicrobials themselves. Indeed, only 2 years after the first arsenic-based drugs became used for sleeping sickness treatment, specifically in colonial contexts where they were used on a mass drug administration basis, clinicians started to report cases of so-called drug fastness. This means that the strains were no longer as susceptible to the use of the antimicrobial drug as they had initially been. And you can actually see this here on this admission form from the Robert Koch Institute, where a readmitted patient where the strain of a readmitted patient is becoming increasingly resistant to ever higher doses of actually quite highly toxic arsenic-based medications. And by 1916, this phenomenon of drug fastness is no longer seen as something that's isolated or, that's a curiosity. It's actually being subjected to systematic research by microbiologists. And by the 1920s, concerns that this phenomenon is becoming so widespread, for example, already lead to a change of drug treatment treatment protocols by the colonial and international sleeping sickness campaigns during this time in Africa specifically, and by the 1930s and 1940s, are already leading to calls to use especially sulfonamide drugs rationally or in a more targeted fashion, well before, again, and I have nothing against Fleming personally, but he clearly was not the first person in 1945 to warn about AMR. Again, this was actually already quite established science during this time. So, why are people using antimicrobials with such an abandon, as we might say from our perspective now, if they knew all about AMR? Well, in hindsight, perhaps two factors are perhaps most prominent to explain this when we look back at this early bonanza. The first was because of this systematized belief in big science, big industry, big states to solve the world's problem. There was a confidence that science and human ingenuity would be able to keep up with microbial evolution. And the second and perhaps more important reason was also that the knowledge of AMR that existed during this time was a knowledge that was not yet completely based on modern understandings of bacterial genetics. By the 1940s, there is a conception that bacteria are subject to modern evolutionary theory, so they're no longer based on Lamarckian resistance acquisition, but there's still the belief that resistance occurs as a result only of random mutations, and as a consequence, it's enough to apply infection prevention control strategies to prevent the spread of an individual strain proliferating, and that you're not really dealing with an ecological phenomenon, which enables also us to understand why, for example, during this time, people are so unconcerned about animal antibiotic use and antibiotic exposures in the environment, because they think that, well, if we ever have a resistant strain cropping up, we'll be able to deal with it just like we deal with any other kind of surging bacterial infections. We'll just switch around the antimicrobials, and also we'll use traditional containment protocols to get on top of it. So we're not really dealing with a planetary challenge here yet. This knowledge, however, does gradually start changing during the 1960s as a result of Japanese researchers in the late 1950s discovering resistance factor transfer, so the fact that resistance can be communicated, especially in their context, via the exchange of small extra chromosomal fragments of DNA called plasmids, via horizontal gene transfer. And so by the 1960s, once Western researchers have first translated these papers and then also start conducting systematized laboratory research of their own, British microbiologists, specifically here Ephraim Solar Anderson, based at what was then the Public Health Laboratory Service in the UK, start warning that genes have an epidemiology of their own, meaning that AMR is no longer just preventing the spread of an individual superbug, but is actually something that is proliferating at a genetic level globally, and as a consequence requires new forms of interventions. And these calls are taken so seriously, actually, that in 1966, the New England Journal of Medicine is already warning of a return to a pre-antibiotic Middle Ages. So very similar, actually, to the kind of initial Dame Sally Davis reports from the 2010s. And the New York Times is describing AMR as a threat to spaceship Earth. And by the 1970s, it leads to increasingly contested regulation of antimicrobial selection pressure in human medicine and the agricultural systems. But here, regulators and also clinicians start facing a dilemma. If every use of antimicrobials can select for antimicrobial resistance, how strict should stewardship protocols be? I mean, which aspects of stewardship, which aspects of all of these useful antimicrobial infrastructure should we leave intact? And should we forego some benefits altogether? And there's really no global consensus about how to move forward at this point. And indeed, what happens instead of a kind of globalized or international commitment perhaps to phasing out this rising selection pressure on the microbial commons, what happens instead is a kind of series of what a colleague of mine, Claire Chandler, has described as technical quick fixes, so that you've got numerous public health interventions trying to target AMR from the 1950s onwards, but none of them are really addressing the structural drivers, the underlying infrastructures. And these interventions, they range from practitioner education. So from the 1950s, if you're training in medicine, you'll be educated in rational drug use, so to not use antimicrobials to cut down on diagnosis time. Patients and farmers are being told not to be pushy, so to demand antimicrobials unnecessarily. I mean, what does unnecessarily actually mean from a private perspective? Who knows, right? Especially in resource poor settings. There's also growing investment in infection control in hospitals to curb the spread of the first antibiotic-resistant pandemic, Staph 8081. And by the 1960s and 70s, there's also increasing investment globally in standardizing AMR surveillance. You can see here these MIC figures from the 1960s, the minimum inhibition concentration charts, as well as the first push through of antimicrobial restrictions of limited amounts of highly relevant therapeutic antibiotics, such as penicillin and the tetracyclines, in parts of animal production in Europe. And from the 1980s onwards, there's also focus on antibiotic innovation. But none of these measures are really integrated. There's no underlying driving global action plan here that's going forward. And also, none of these interventions are really actually addressing the factors driving antimicrobial usage and dependency. So the fact that some healthcare systems just can't afford really to restrict and steward antimicrobials in the way that some of the more well-resourced can do, or the fact that some farmers are no longer able to compete without relying on antimicrobials to keep infections down in the setting of increasingly low profit margins. And perhaps from my current research perspective, most importantly, there's also no focus beyond the farm and the clinic towards the fact that it's not just the pharmaceutical use in the clinic and on the farm that's driving antimicrobial resistance selection, but actually increasing selective pressures outside of clinics and farms, for example, in the context of factory effluent. So every antibiotic that has ever been used has been produced in a factory, and these factories often release extremely high levels of antimicrobials into the environment. And there's also no focus really on what happens once antimicrobials have been secreted out of a system. So once you've swallowed an antibiotic, it's been affected. Lots of antibiotics still have breakdown products or actually get excreted in still pretty much intact ways into the environment where they continue to be selectives. And this is despite the fact that from the 1970s onwards, microbiologists are increasingly warning that there are significant AMR reservoirs building in soil and water. And just to give you an idea of this, for example, I think China these days produces around 400,000 tons still per year of fermentation wastes to produce biological antibiotics. They produce around 80% of all active pharmaceutical ingredients in the world. And this is just nowadays. So if you think about this back to the very early days of the kind of mass antimicrobial era, you can think of the hundreds and millions of tons that humans have released into the environment without ever really paying attention to what these substances are doing here. And I've riffed this slide off a colleague of mine, Will Gaze, in the United Kingdom, who in the 1970s already finds microbiologists employing pretty much a One Health triad figure here to show that urban, agricultural, environmental settings are all linked. And yet, this isn't really captured in any of the global action plans or international plans that are being designed to coordinate action on antimicrobial resistance. Indeed, even when these international action plans start arising, with the first WHO call to action published in 2001, and then in 2015, the WHO publishing its global action plan, the focus is still very much on the clinic and to a very limited extent on the farms. The focus is still on trying to standardize all of these technical quick fixes without necessarily addressing underlying structural drivers. And this is both due to the fact that high-income countries during this time are realizing that their own stewardship protocols are not enough to deal with AMR, but that actually AMR is a global problem. And it's also linked to the fact that certain high-income countries are trying to show leadership during this time, most notably the UK under David Cameron, seeing AMR as a foreign policy area in which the UK can show leadership. However, the export of the high-income country protocols that again focus mostly on farms and clinics during this time are not necessarily based on straightforward correlations between reductions of antimicrobial usage and AMR reductions. And again, most of them leave the wider drivers of antimicrobial usage demand by patients and farmers unaddressed, which means that when you export them, it often becomes a game of blame. Who's responsible for which level of antimicrobial usage? And if you read policy reports during this time, what you increasingly see is that countries who cannot comply to the technical fix system that's been devised mostly in high-income countries increasingly get blamed for causing AMR at a global scale, even though historically speaking, they've had the least access to antimicrobials. And so as a historian, I'd say that AMR becomes very much subject to the wider securitization of global health discourse during this time, where global health problems are primarily made sense of as biosecurity challenges for the high-income countries to deal with a kind of command and control form of surveillance and auditing, without necessarily really addressing the underlying drivers of both usage and also increasing resistance resulting from stressed microbial environments. And perhaps unsurprisingly, this kind of patchwork attempt that then gets bundled together with the 2015 action plan hasn't been very successful in curbing AMR rates, or hasn't been, and also AMU rates. Indeed, if we look at the most recent Lancet effect, calculation of AMR, which is an extrapolation of AMR burdens from the 1990s to 2050, the Lancet sees AMR continuing to grow, specifically in populations 5 years and older around the world. Antimicrobial usage is still predicted to rise around the world, again, because antimicrobials are so key to developmental promises that it's very difficult actually to tell middle-income countries and low-income countries to stop using them if their use brings all of the benefits of the modernity, which is the goal of developmentalism at the same time. And there's also been growing concern about the fact that high-income countries' reductions of antimicrobial usage that have been achieved since the 1990s are still not high enough the reductions haven't been strong enough to actually get below levels at which antimicrobials were already believed to be selective in the 1960s. So even if all of the world were to emulate HIC's success, for example, in cutting antimicrobial usage in veterinary sectors, for example, the level of reduction would still not be enough to get us down below levels in the 50s and 60s when the effects of antimicrobial resistance selection were already becoming visible. And finally, the investment in innovation has so far not resulted, in a sustainable pipeline of new molecular compounds to deal with AMR, and perhaps most worryingly, they are still mostly focused on high-income markets and not so much on low-income and middle-income needs, where AMR burdens are the highest. The failure of the Global Action Plan, despite a lot of hype around it in 2015, to result in meaningful success in accordance with its own key performance indicators over recent years, has also led at the international level to a declining interest for AMR. In 2021, we performed a survey of international reports by the WHO on AMR since 1948. And what we saw that after reaching a high point in and around the years surrounding the Global Action Plan, reporting numbers actually started declining. And this is also matched if we look at investment figures captured by Germany's Global AMR R&D dashboard, where we see, following a high around 2019, 2020, perhaps also correlating with the COVID-19 pandemic, an ongoing reduction of AMR, AMU. And in order to regalvanize attention for AMR, the UN General Assembly only last year tried to create a meaningful, almost carbon-like approach, climate change-like approach to AMR by saying that we need a standard global goal of a reduction of 10% of AMR deaths by 2030 when measured against the 2019 baseline. And it once again called for a focus on stewardship, infection prevention and control, water and sanitation and hygiene, and access and innovation. So very much still kind of repackaging these technical fixes from earlier, and including this time also an explicit focus on One Health and clinics and farms. And now the question is really whether any of this will actually result in meaningful change, right? I mean, as I've shown, we've been in many ways repackaging the same policy solutions for 50 years now, and so far the impacts have been limited. So is the solution really to go harder, better, faster, stronger? You know, I'm in France, so I like to quote Daft Punk. Or are we actually missing something with our approach? And to kind of start the final part of the presentation today, I'd like to turn my attention to the area that's so far been missing from global policy discourse about AMR, which is the environment. Indeed, in the UN General Assembly report, it's only mentioned 3 times in 106 paragraphs. And the word microbes, right, the thing that is actually reacting to antimicrobial selection, the thing that is then causing problems for humans in clinical and agricultural settings, is not mentioned once. And indeed, this is entirely in keeping with international trends, where only 2 out of 248 international policy of documents on AMR exclusively targeted the environment since 1945. So still in the global policy discourse, targeting or stewarding antimicrobials rather than microbes remains the priority, right? The clinic and farm still dominate our imaginaries when it comes to thinking through AMR, and we don't really see AMR as perhaps signaling something wider about the state of the microbial commons around us. And this is the question I'd like to pose to you in the final 9 minutes of the presentation here, which is what happens if we start reframing AMR not just as a clinical threat, but as an environmental phenomenon. So in other words, what happens if we see AMR not just as the failure of a drug to work in a specific setting, but actually as something telling us something much more broadly about the state of the microbes that surround us and on whose functioning we depend for our lives and livelihoods in many ways. And with this, I turn to the Antibiocene project, a project that I'm starting with colleagues of mine, where we see AMR not just as a clinical threat, it undoubtedly is causing lots of death and misery around the world, but also as a biosocial signal of a more longer-term genetic shift of the global microbial biosphere caused by accelerating pharmaceutical and non-pharmaceutical selection, right? So what if AMR here is a signal very similar to, rising temperatures associated with climate change, of rising stress in the microbial world? What would that do to our global governance frameworks? And also, what would it mean if we actually take AMR seriously, not just as one global phenomenon, but actually as something that is very different in different areas around the world? Recent metagenomic surveys strongly indicate that AMR is not the same everywhere across the world, but is actually associated with distinct forms of anthropogenic stresses in different environments, which can range from climate change, biodiversity loss, but also to the availability of certain antimicrobials in certain zones, but also exposure to other pollutant selectors. And it's also something that we can study not just within the present, but as something also that we can measure through time. So the antibiocene is not only something that is global, but has distinct biogeographies, but also something that is accretive and something that has been accelerating since the beginning of the 20th century, perhaps even since the 19th century, with measurable spatiotemporal golden spikes of antimicrobial resistance genes still measurable in soil cores and lake sediment data that we're generating from the environment. And some of these soil core archives, for example, have been analyzed in the context of the Netherlands, where they find a distinct rise of antimicrobial resistance genes, for example, associated with the mass introduction of tetracyclines into Dutch agriculture. So what does all of this mean then for how we might approach AMR? Well, in our opinion, it opens the way for a new biological and social sciences informed approach to studying AMR as a way not just to steward the antimicrobials, but perhaps also to steward the microbial commons more widely. And in this, we draw on the work of a social scientist called Hannah Landecker, who's encouraged us to think of a microbe as a socio-ecological event, right? A microbe just doesn't just exist in kind of empty space, but actually a microbe exists within complex communities that are also shaped by human communities. And we can study this both biologically, if we look at the accretion of AMR in different bacterial species over time. So we can look at, in the case of Salmonella enterica zirova typhi, so typhoid, an organism which I've been familiar with for quite a while. We can see how AMR resistant, so resistant strains associated with the spread of a new haplotype, H58, have not only spread globally, but have actually displaced sensitive strains of typhi in different habitats. We can look at accretion of ARGs over time, so soil cores, but also lake sediment data show us that AMR rises, usually associated with distinct anthropogenic impacts, such as pharmaceutical and non-pharmaceutical selective substances. But we can also see how the mobilome, so mobile genetic elements, which convey genes across different microbes, have also changed as a result of the selective pressures that we've brought to bear on them as a species, with, for example, sequencing of the Murray collection in the in the US, indicating that those aspects of the mobilome which have survived to the present are strongly associated with the conveyance of resistance genes. And we can link all of these biological dynamics also to social dynamics. So we can then see not only how does AMR accrete, not just in the clinic and the farm, but also how wider AMR and selective pressures on microbial environments might also drive forms of societal stress in human societies. So what happens in the long term if you live in a highly polluted environment, let's say downstream of an antibiotic manufacturing plant in India, as opposed to people living on brownfield sites that are currently being repurposed in the UK and the US, which formerly housed antibiotic production. Do these environments lead to specific stress on healthcare systems? Do people have specific health effects? Do certain industries such as dairy and breweries suffer long-term effects? And also, can we start linking effects of microbial stress to broader forms of human stress, for example, in the case of war, where the selective release of lots of heavy metals and biocides into certain environments might drive resistance selection, leading to higher forms of antibiotic usage by humans, leading to more resistance. So can we start thinking of AMR not just as something that is limited to the failure of a drug to work in a certain setting, but actually as something that signals a wider disruption of microbial environments and commons? And then more importantly, can we identify what forms of microbiota, what forms of microbial commons it might be worthwhile to protect, stabilize, and what forms of dysbiotic dynamics to avoid moving forward? So, to come to an end of this kind of whirlwind tour through the history of antibiotic innovation, antibiotic use, resistance, and antibiotic regulation, and then the kind of turnaround we're foregrounding the environmental perspective here. What I hope to have shown you here is that AMR is not just a kind of current global health issue, but it has actually occupied regulators, clinicians, scientists, veterinarians for well over 120 years, and that there is a value here in starting to foreground middle term and also long term, longue durée conceptions when it comes to approaching it. By linking long-term perspectives on AMR also with a kind of environmental reconceptualization of AMR, what we start seeing is a problem that will not be tackled just by focusing on patient behavior or farmer behavior, but requires much broader structural interventions into the infrastructures that we have created to drive healthcare and food production around the world. Tackling AMR will mean systems change, and that will likely be expensive, and it will likely not result from just interlayering lots of different forms of technical fixes. At a broader ecological level, AMR also, if we take it seriously as a planetary phenomenon, encourages us to think of microbes as part of the planetary boundaries. Are there safe planetary boundaries we should stay in as a human species when it comes to stressing our microbial environments? And what are these boundaries? And what also are the microbial environments that we might want to preserve, stabilize, manage? I'm still looking for a good word to describe this. And finally, it also attunes us to the fact that AMR stewardship is not something where individual responsibility is equal across the world. Large parts of the global population have never had sufficient access to antimicrobials in the first place, but have been disproportionately at the same time impacted not just by infectious disease burdens, but also by pollution burdens. So, can we ask these populations to bear the same burden of stewardship as richer parts of the globe? And also, legally, is there perhaps a responsibility for industries to not just clean up toxins and carcinogenic impacts and externalities resulting from production, but also dysbiotic long-term effects? And I think with this, I'll leave it and open up to questions. So thank you very much.
Thank you so much, Claas, for this very vast and inspiring talk. And it's actually quite impressive to see how many different aspects of you mentioned planetary boundaries, and it's really true that you have to take everything into account. It's really impossible to focus and implement interventions on one sector and ignore another. This really comes down to the one health aspect that needs to really be taken into consideration for any kind of work plan. And thank you so much for this talk. And I am wondering if there are any questions. Okay, so just to remind everybody, you can either raise your hand and ask the questions once I allow you to unmute yourselves, or you can ask them in the chat. So we have a first question in the chat box. What do you think of bacteriophages, and how do they fit into a potential strategy to curb antimicrobial resistance?
Thank you. Yeah, I'm a big phage fan, in the sense that I work a lot of them in another aspect of my research. Now, I mean, phage predation obviously also drives a lot of microbial evolution, and the dynamics of phage predation and also antimicrobial selection pressures, I think, something that is interesting a lot of microbiologists these days. Bacteriophage therapy has obviously been around for pretty much as long as the antimicrobial era has also been around. So it starts in the 1920s, and it becomes very popular in the 40s and in the Soviet Union until the 1980s. Um, I've got a paper I'm working on at the moment where I trace the use of phage therapy in France until the 1990s. And what's quite interesting there is that phage are at best a complement to existing antimicrobial therapy. They're never really an ersatz. So phage aren't the next magic bullet or, the next wonderful substitute of antimicrobials that will somehow fix and get us out of the dilemma of antimicrobial resistance. They're an incredibly useful tool when you're dealing with chronic multiple drug resistant infections. But I think the thing with phage is very much the same thing with antimicrobials, is that if we use them to simply plug gaps in our antimicrobial infrastructure, so all of the selective pressure that we're bringing to bear on the microbial commons of the planet, we may just end up replicating the same problems. We'll still be driving resistance evolution. We'll still be putting pressure on the microbial commons around us. The advantage that phage do offer, however, is that they're very tailored, that they're very specific to specific settings. So with phage therapy, how it's really worked both in the West and in the East over the last 50 to 60 years is that they are a very useful complement for a very specific section of patients. So I'm both a great believer in the fact that they do allow us to get away from using ever higher doses of antimicrobials for certain patients, but they are not a wonder solution to AMR or the antibiotic dependencies we've created.
Thank you. Unless, of course, we can design a phage that stops microbes from evolving altogether, but that would be...
Yeah, but I mean, do we even want that? I don't think we
No,
really do,
probably
right?
don't. We
No, we want
don't.
to have a sustainable relationship.
Yes, indeed. Next question. Why do you think UN policies are not mostly targeting AMR in the environment? Is it because collecting metadata and environmental samples is challenging?
Yeah, I think on the one hand side, it's the environment is much more complex than the clinic, and or the farm for that matter. So I think it's definitely to do with data collection. I would, however, also say that it's to do with prestige within microbiologist circles and also professional associations, where regardless of when you look, uh, environmental microbiology has not been the most prestigious aspect and not the most influential aspect of the microbiology community for a very long time. So I think it's also to do with the fact that even though there was solid data for certain externalities, if we look, for example, at pharmaceutical effluents, for a very long time, this community was just not listened to. It was endlessly drowned out by calls for action in other areas. So that's one aspect of it. I think the other aspect of it is that politicians often historically have liked easy targets with key performance indicators whose success you can manage and measure within 5 to 10 years. And those are easier to propose if you're looking at a metric like antimicrobial usage or perhaps resistance within an individual clinical setting than it is to do with the complex dynamics of AMR within a wider environment. And I think this is why in the final part of the talk, I think I see AMR as a signal, but not necessarily as the only signal for wider microbial stress in the environment. I think it's time now, and I think lots of microbiologists think this way, to start considering the broader state of the microbial commons. And I think AMR is a great way in terms of the historical data that we already have of measuring change over time. So yeah, and perhaps the other thing is also um, rivalries between health organizations themselves. If you look at government ministries, you will rarely see an environmental ministry that is resourced to the same extent as the health ministry or the agricultural ministry. And you will rarely find a health ministry that is willing to hand over control and resources to another ministry in an overall cash-strapped setting. And it's not very different at the international level either. So WHO had its stamp on AMR from 2001 onwards, and it's telling that UNEP, the UN Environmental Programme, was only admitted to what then became the quadripartite 20 years into this discourse, even though the data was already mounting before then. So politics, scientific prestige, and data difficulties all kind of come together.
Okay, thank you so much. So there are further questions in the chat. Some of them you have already targeted in some of your responses. There's one on the WHO, actually, that says WHO highlights that many people worldwide lack access to essential antimicrobials. How can we balance efforts to reduce antibiotic overuse with the need to ensure access in low-resource settings where different factors drive AMR?
I think that's obviously it's an unanswered question how this balancing act happens. Be it in HICs or globally, where you have in many low-income countries no access to any kind of reserve antibiotics that we would need in order to comply with, for example, aware categories by the WHO. I think these dynamics of trade-off need to be made much more explicit in the policy discussions around AMR. So if you read the UN General Assembly report that came out in 2024, it suggests that we have the solutions to AMR. It suggests that if only we do these things and if only we kind of invest in all of these things, we'll kind of somehow come to a goal. It doesn't really make explicit the power dynamics involved in prescribing who should have access to which drug at which point and whose interests are more important than those of others. The case of colistin is a very interesting one where colistin had been essentially phased out in the 70s and early 80s by lots of high-income countries because of toxic side effects. And then they were rediscovered as reserve antibiotics once the more expensive, less toxic antibiotics started becoming less effective as a result of resistance, leading then in 2015 to lots of calls for stewardship in settings that had come to use colistins because Western pharmaceutical companies had started selling them there as animal drugs because they were no longer high-priced drugs that could be sold in Western markets. So these historical dynamics and trade-offs and who's using what drug and whose priorities should dominate are almost never made explicit at the international level. I think we're much more advanced in the carbon space, where poorer countries are saying richer countries have used more carbon historically. So we're dealing with an imbalanced playing field from the beginning, and I think this needs to be much more explicit in the AMR field.
Okay, coming back to the UN General Assembly, there's a question regarding this, and it says, do you think the UN General Assembly is the right international coordination mechanism, or is there another mechanism that has been proposed, UN or not, which could better focus attention on the drivers of AMR? I guess you think if there are better proposals and work plans out there.
I mean, I think the last 4 weeks since the new presidency of the US have really shaken up the international organization system in general. I mean, technically speaking, the UN level would be a great level to have an open discussion about AMR, or at least have an inclusive discussion that brings on board nations from all across the world. In practice, the UN General Assembly, it's a good place for raising awareness. It's not necessarily the place where global action has happened. Do I see another forum at the moment? Probably not. I don't think we have a better forum, and I think even the fora, the very limited fora that we have, are under significant pressure these days from the global turn away from multilateralism. So I think where I see the future of lots of these stewardship policies lying is probably at the moment more at the regional level and at the national level, in the context of the EU, for example, but also in the context of the African Union, for example, in terms of deciding what forms of access and innovation priorities best serve regional needs. The UN system is a great way of finding consensus positions, but I think the meaningful action that we are seeing and also seeing in middle-income countries like China, for example, with strict environmental regulations, seems to be the one that is more dynamic at the moment.
Okay, thank you so much. And then now a more personal question. What one policy would you put into EU law were you given the opportunity?
Um, I think that what the EU needs to have, and this is just purely my own research interest, so feel free to shoot me down, but it's quite remarkable we don't have any EU standardized guidelines for AMR in wastewater. We have guidelines for the Urban Wastewater Directive, certain things are now coming in, but we've really neglected microbial dimensions in pharmaceutical effluent regulation, for example. And it was very interesting if you've been following the debates there that it was actually India that was really pushing for statutory regulation before they ran into difficulty as a result of industry opposition in, I think, 2018, 2017. That's when these discussions emerged, and that the debates were then only taken up in the European context. So I think in an EU context, I'd like to have a much stronger focus on effluent regulation with regards to selective inputs.
Okay, and this fact of industrial resistance, I can somehow understand it. And I was wondering, you mentioned that low middle income countries haven't really yet seen this kind of developmentalism that higher income countries saw 70 years ago, thanks to antimicrobials that were structural in making this happen. So isn't it in a way somewhat unfair that these low and middle income countries haven't had this opportunity to catch up using that same infrastructure?
Yeah.
And well, I guess the answer is yes, it is. But
It
at
is.
the
I mean,
same,
it is an unfairness, right?
yeah.
And it's in part the market was always structured, and also if you look at modern intellectual property laws, um, the market that pays the most to develop for is traditionally the US, followed by European markets, and as a consequence, these markets see the greatest benefit of novel compounds introduced before these compounds become available in other parts of the world. Ergo, also before resistance starts becoming a big issue. So it's the system is stacked. Innovation wise, definitely against rebalancing this already strong historical debt going forward. I mean, it's another work stream I'm working on, which is antibiotic innovation policies. Um, it's it would be very interesting to conceive of an innovation system that perhaps doesn't prioritize molecular novelty as much as access considerations and repurposing of older drugs for new applications in low and middle income settings. And so perhaps rebalances some of the innovation work and subsidies that are primarily still serving high income patients towards lower income populations. That would be a nice thing also to see going forward. But because much of the innovation policies we have currently are designed to fix markets, we're not really questioning the historical imbalances that have for a long time made the newest drugs available to the richest people. And then when they become less effective, that's when they become available to other people too.
I think this is an important point, which is shifting away the attention from molecular novelties, because they're not always the solution, right? We have a comment in the chat from somebody from Ines saying she's working on antimicrobial peptides as a potential alternative to antibiotics, but it's very challenging because they have seen that even antimicrobial peptides can also lead to resistance. So, of course, once there's evolution, resistance will always be a factor, right?
I think to kind of so I'm part of a group that's called Just Transitions with the British Academy, and if you think of antimicrobials as a scalar technology, so as a technology you can use anywhere around the world, and you get your microbial control, and that leading then to a global problem of antimicrobial resistance as a kind of microbial reaction against everything being treated with antimicrobials for it to work. Um, I think any technological solution to AMR is bound to run into the same problems because at the end of the day, there seems to be a demand for a technology that just slots into the place where we then kick antibiotics out. So they also become scalar by default. So in many ways, perhaps the solution to AMR is to invest in descalar localized solutions for optimizing microbial environments in specific areas rather than hoping to find the next tetracycline out there or the next bacteriophage or peptide-based technology. I think they all have a role to play going forward. But in many ways, I think we need to get away from this very 20th century notion of the magic bullet, the magic bullet solution to all of these things. And by the way, I read in the chat the note about Hatta. I will definitely correct that in in the slides going forward.
Yes, thank you for the correction comment. It's been taken, it's been noted. And okay, so I think we can wrap up this webinar. I just have one final question. It's more of a historical point. It's really interesting that AMR was reported extensively as early as 1916. And you said one of the problems with it was there was strong belief and confidence in big science, as well as there was a more organism-focused approach rather than considering horizontal gene transfer. Do you think there was also an element of psychological resistance, like people just closing their eyes and ears to the fact that it's something that is so productive and useful to society? We're just going to ignore the problems. And were there kind of like no vaxxers, no AMRs at the time or something like that?
I mean, I think we could ask the same question to somebody smoking a cigarette or, drinking a beer. That's, about the long-term effects. But I mean, as with any technology, you have benefits and downsides going in. And I think for a while it just seemed that these benefits were so incredible going forward. And I mean, they still are so incredible. If you think about life expectancy, cheap meat, cheap plant production, etc., going forward, that it's very difficult to face the fact that it's endlessly self-undermining and also that we might be dealing with externalities which impose ultimately costs that are bigger than the gains that we get. And I think that's a very human problem of tackling with any, tackling any long-term wicked challenge.
Exactly. Well, thank you so much once again for your time and for this wonderful talk. I'm going to wrap up this session right now. And just to answer some of the questions, the talk has been recorded, and I will send you the link to the recording to all the people who were present. My apologies, I didn't start the recording immediately. It was started like 1 minute in. So it skips your index of what the
You
talk is
didn't
going to
miss
be
much, yeah.
about, but yeah, the essential content is there. Thank you so much for everybody who participated, and thank you once again, class.
Thank you very much for having me. And thanks for the great questions. Bye.
Bye.